As mentioned, my name is Sejad Ayyoubi. I'm a pharmacist and PhD candidate at the Erasmus University Medical Center. And at the Erasmus University Medical Center, we are very eager to really bring this technology to the clinical setting. A lot of concept studies have been shown. And in my work, I would like to present a translation from the pharmaceutical 3D printing technology to a clinical setting. So today I will be presenting my research around 3D printing of personalized cortisol for patients with adrenal insufficiency. In my I've divided my presentation in three major parts. First of all, I will give some background on the unmet medical need, so around adrenal insufficiency. In the second part, I will talk about the pharmaceutical development, where we've developed the 3D printed hydrocortisone using fused deposition modeling. Here on the right side, you can see that we can build very complex structures with this technology, and I will later on come back on how we can use it to also build complex pharmaceutical products. And I will finalize my presentation with some remarks on the clinical trial that we have planned at the end of this year. So starting with the background on the disease is very important to know, because later on I will explain why the products that we make are having an added value for these patients. So normally in healthy individuals, the cortisol production is regulated by the HPA axis, which you can see here. So the hypothalamus, pituitary gland, and the adrenal gland. But patients with adrenal insufficiency, they do not produce their own cortisol anymore. This can be due to several reasons, which are stated here. I will not go deeper into these reasons. But take-home message is these patients cannot produce their own cortisol anymore. And this comes with several symptoms that you can see on the right side image here. Some of the symptoms are non-specific, like low blood pressure or gastrointestinal complaints. But when cortisol levels drop to a very low concentrations, patients can go into an adrenal crisis where they develop convulsions, hypoglycemia, hyponatremia, which may be life-threatening. So as these patients do not produce their own cortisol, they are dependent on lifelong hydrocortisone suppletion. And the standard of care for these patients right now, I will dive a bit deeper into the graph, which is shown on the right side here. In this graph, you can see on the y-axis the serum cortisol plasma concentrations, and on the x-axis you can see the time starting around 6 a.m. And the black line is the cortisol plasma concentration in healthy individuals. As you can see, it starts high in the morning, and it gradually goes down during the day and going up again at nighttime. So the treatment goal for patients who do not produce their own cortisol is to mimic this black line. And in the same graph, you can see the cortisol plasma concentration of patients who take thrice daily hydrocortisone, which is the standard of care. They start with 10 milligrams in the morning, 5 milligrams at midday, and 5 milligrams in the afternoon. And the standard of care is suboptimal. This has two reasons. As you can see, it does not overlap the physiological cortisol plasma concentration, which is the treatment goal. It has peaks and low cortisol levels, which is associated with negative long-term health outcomes, such as weight gain, diabetes, osteoporosis, but also a higher cardiovascular risk. And the second disadvantage of the standard of care formulations is that the nighttime cortisol is not treated, which means that patients wake up very ill in the morning and they take their first hydrocortisone pill, which is this 10 milligram pill, and they have to wait until it kicks in to start a normal day. So these patients have a very poor quality of life. And the second formulation that is registered for these patients is the plain, which is a very expensive hydrocortisone formulation. Here you can see the same graph But now you see the black line is the plain adren. It's a sustained release hydrocortisone formulation. As you can see, it produces more stable plasma concentrations. So it's a bit better than the standard of care, but it's still suboptimal because it does not take into account the nighttime cortisol, which I've shown before. And it's also available in fixed dose formulations in 5 milligrams and in 20 milligrams. While there's a very high variability in the cortisol need per individual patient. So think about, for instance, patients who are older and younger, but also patients who work out more, they need more cortisol compared to patients who do not work out more. And there are many factors influencing the cortisol need for specific patients. So what you need is to actually tailor the cortisol dose, hydrocortisone dose per specific patient. And the last disadvantage of this formulation is that it is not the standard of care due to several reasons. One of the reasons is that it is not insured by the healthcare insurance in the Netherlands, even though that it produces more stable plasma concentrations. So in the first part, I've explained that we have a group of patients who do not produce their own cortisol. And I've explained that there are 2 registered cortisol formulations, which are suboptimal at this point. So the question is, how do we want to improve the treatment, pharmaceutical treatment of this patient group? We have the idea to develop 2 new hydrocortisone formulations, 3D printed. First of all, we have the sustained release formulation, which is for the daytime cortisol, as you can see in the graph below. And second of all, we have the nighttime cortisol formulation, which is the delayed release formulation. And the delayed release formulation is taken in before sleeping time. The shell will erode, after which in a couple of hours, the hydrocortisone inside will be released, producing the rise in cortisol at nighttime. So the added value of these formulations is that we take into account the nighttime cortisol, which is not taken into account by the registered formulation. And we have a daytime sustained release formulation, which we can tailor. We can tailor it based on the tablet height. And I will come back to that on in a later slide. And the third added value is, of course, that we have a better therapy adherence because we dose twice daily instead of the thrice daily of the standard of care. So we started our pharmaceutical development starting with the aim of developing the sustained release 3D printed hydrocortisone formulation. Based on literature, we selected a few polymers. We knew that the hydroxypropyl cellulose and the A2 cellulose, for example, produce a sustained release effect. And we've developed 2 formulations with 2 different hydrocortisone concentrations. I am not sure if, I think a lot of people are familiar with the FDM printing process, but nevertheless, I will go very fast through explaining this. So we have the raw material, which we pre-mix and put into the hot melt extruder, after which we obtain the API-loaded filament, which you can see in the image below. We've chosen for a design of 6 millimeters, a tablet design, and we've added the design and the filament into the FDM 3D printer, after which we obtain our personalized hydrocortisone dose. This is a bit of trial and error, but we've chosen a printing temperature of 200 degrees and extrusion temperature of 170 degrees. And we've chosen to print 3 different tablet heights in order to assess the effect of tablet height on the dissolution profile. And actually here on the right side of this slide, you can see the very first hydrocortisone tablet that we've printed, I think 2 years ago. As you can see, it is a bit discolored, it's a bit brownish, which may indicate that some extent of the API has been degraded. I will get back to that on a later phase. So with the 2 formulations that we've developed initially with the 3 different design heights, we performed a dissolution study On the y-axis, you can see the hydrocortisone mass in milligrams, and on the x-axis, the time in 24 hours. The blue line is the sustained release plain adrenal formulation, which is a registered formulation, and the black lines are our 3D printed formulations. As you can see, we are not only able to almost mimic the plain adrenal formulation, but we are also able to produce the same sustained release effect, but with different doses. For example, you can see here 12.7 milligrams, 7.6 milligrams. So we've shown that we can produce sustained release effect while maintaining with different doses. But still, there's some room for optimization, as the European Pharmacopoeia recommends a drug release of 80% within 24 hours, and our tablets did not comply to this. So this was the first step that we had to optimize. The second thing that we did was we wanted to have an easy way to adjust the dose. So we did an experiment where we adjusted the tablet height starting from 0.5 millimeters to 3 millimeters, and performing content analysis of the hydrocortisone. And as you can see in this graph, there's a very high correlation between the tablet height and the hydrocortisone contents, which means that if I get a prescription from a doctor who wants to print 9.5 milligrams, I know that I have to print around a tablet height around 2 milligrams to obtain these doses. So this is a very easy way for us to play with the tablet height to obtain the doses that is prescribed for the individual patient. And lastly, what we did with this formulation was to do a content analysis in order to see how much of the hydrocortisone is left after the manufacturing process. As you can see here on the right below side, at the printing temperature of 200 degrees, we only had 71% hydrocortisone left. So this was the second point that we needed to optimize in order to get a higher hydrocortisone content to minimize degradation. So we've identified that some degradation may have happened. So we designed a quality by design study, in which in this table on the left side, you can see the temperatures range starting from 200 degrees going to 150 degrees. And at every temperature, we did content analysis. As you can see, the lower the printing temperature, the processing temperature is, the higher the average content of our tablet is. So indicating that we've minimized the degradation that we've observed initially. You may wonder where is the remaining 8%? We think that the remaining 8% is lost. Some of you might know that hydrocortisone is a very sticky substance, and we see that it sticks at the mixing process and at the extruding process. We still have to back this up with evidence, but at least we have shown, we can also visually see that. We have shown that degradation is minimized. Here below, you can see the very first tablet that we've printed at 200 degrees. And if you compare it with the tablet that we've printed at 150 degrees, the last one is very white and it looks far better compared to the very first tablet. So we moved on with this printing temperature, and we've changed the formulation a bit, adding polyethylene oxide and Hydrojet RL in order to optimize also the dissolution profile. And the last thing that we changed in the formulation, in the design, was that we went from full tablets, which you can see on the right side, to donut-shaped tablets in order to optimize the dissolution profile. And we've performed a dissolution once again. And here in this dissolution graph, you can see two y-axes. The right orange y-axis corresponds to the orange lines, which is from 2 batches. As you can see, we managed to have a drug release of 10 milligrams in 24 hours with the final formulation. And if you take a look at the blue y-axis, which is the hydrocortisone in percentages, which corresponds to the blue lines of 2 batches, we've managed to comply to the EP recommendation of drug release of a minimum 80% in 24 hours. So actually, this is the formulation which we want to take into the clinical trials. Of course, we need to do a few more experiments in order to set up the production process. But before going to the clinical trial, there are a few other experiments that are very interesting to present today. So we have an FDM printer in which we can choose the resolution, which we can set the resolution. We can set it at low, standard, high, or hyper. We were very curious what the resolution does to the average content of the hydrocortisone we recover. As you can see, the lower printing resolution we choose, which translates to a lower end quality of the end product, we get a higher hydrocortisone content. This is due to the fact that the API is the lower the resolution, the faster the printing speed, which means that the API is for a less time exposed to the high printing temperatures. Therefore, we get these high hydrocortisone contents. The other thing that we did was a variability study. As you may know, the requirement of the EP is below 0.1%, and our tablets comply with 0.06%. And the last experiment that we did with our latest formulation was differential scanning calorimetry. If you look at the above line, which is the raw hydrocortisone, you can see a very clear melting peak. And if you look at the very below line, which is our tablet, you can see that this melting peak is disappeared, meaning that our hydrocortisone is integrated in our end product in an amorphous state. So actually, this formulation I've shown here before, this is the formulation we want to take into the clinical study. What we want to do in this clinical study, it's actually a feasibility study. The question we want to answer with this study is, does our 3D printed 10 milligram sustained release hydrocortisone produce adequate cortisol plasma concentrations in vivo? We've shown that it produces stable concentrations in vitro with our release profile, and now we want to back that up with in vivo evidence. And we want to do this by randomizing 20 patients into 2 groups. Group 1 receives conventional therapy, which is suboptimal on day 1, and on day 2 they will receive the 3D therapy. And group 2 will receive the same treatment, but in the opposite order. And the samples that we want to take are blood samples and blood pressure as a safety marker. And as an endpoint, we want to compare the areas under the curve of the 2 treatment strategies. As you can see on the right side, the graph, which I've shown a few times before, we want to show the same graph with the immediate release formulation, which is the dotted line. But we want to show also in this graph our sustained release formulation. So finalizing my presentation, some future remarks. We are also planning a stability study in order to show that our 3D printed products are stable within 6 months. Of course, we are setting up the production with the Spanish partner of the sustained release product, and at the same time developing the delayed release product, which is for the nighttime treatment, as you may recall. We've already managed with our FDM printer to use 2 commercial filaments to create these combined end products, as you see here on the right side. And for clinical studies, we also want to do a clinical study with the nighttime cortisol that we are preparing, and in the end, a combined randomized controlled trial of the sustained release and delayed release 3D printed products compared with the standard of care. So this was my presentation, and of course, I did not do all this work by myself. We have an amazing team at the Erasmus University Medical Center which I wanted to thank, and also thank the organizing committee for me to be able to present my work today. And there's one more thing I would quickly want to show. At the Erasmus University Medical Center, we are also working with flecainide for children, as Anton mentioned before. And we are also working on 3D printed liothyronine T3 for patients with hypothyroidism. Yes, thank you very much, and I'm happy to answer any questions.